Hello students. So we are going to study apoptosis. Apoptosis is a pathway of cell death in which cells destined to die activate the intrinsic enzymes that degrade the cell's own nuclear DNA and nuclear and cytoplasmic proteins. So it is a pathway of cell death in which the cell is destined to activate its enzymes that degrade the cell's own DNA and nuclear and cytoplasmic protein. It is a Greek word meaning falling off. Apoptosis is a programmed cell death as it is genetically regulated. And apoptosis is different from necrosis in the way the cell membrane remains intact in apoptosis and there is no leakage of intracellular material. And apoptosis does not induce any inflammatory response. Apoptosis, it could be physiological or pathological. Physiological apoptosis. Apoptosis is a normal phenomenon that serves to eliminate unwanted, aged, or harmful cells in physiologic apoptosis. Samples of physiological apoptosis are first apoptosis during embryogenesis, involution of cells, and second is apoptosis due to withdrawal of hormones like endometrial shedding during menstrual cycle, atresia of ovarian follicle, regression of lactating breast after weaning, and prostatic atrophy after castration. So these are examples of apoptosis, physiological apoptosis due to withdrawal of hormones. And third is due to loss of cells from the renewable tissue. Examples: epithelium of intestine and crepe. Now the pathological apoptosis. Cell death in tumors exposed to radiation or chemotherapy is an example of pathologic apoptosis by the activating p53 or tumor suppressor gene. p53, it will stop the cell in G1 phase of cell cycle so that the damage can be repaired. If the damage can't be repaired, p53 causes cell death by apoptosis. Cell death by cytotoxic T cells in immune mechanisms are also example of pathologic apoptosis, such as graft versus host disease and rejection reaction. Cell death in viral infections, example, formation of Councilman bodies in viral hepatitis. The loss of neurons through apoptosis in degenerative diseases of CNS, examples, Alzheimer's disease and Parkinson's disease. Now the morphological changes in apoptosis. The cell will shrink. So cell shrinkage is important. The cell is small, shrunken with dense cytoplasm and chromatin condensation. Most characteristic feature of apoptosis is chromatin condensation. The chromatin aggregates peripherally under the nuclear membrane into dense masses of various shapes and sizes. The nucleus may break up into fragments. There is formation of cytoplasmic blebs and apoptotic bodies. The apoptotic cell first show extensive surface blebbing that undergoes fragmentation into the membrane-bound apoptotic bodies. So, there is formation of apoptotic bodies. Phagocytosis of apoptotic cells or bodies by macrophages. This is the diagrammatic representation of morphologic changes in apoptosis. The normal cell with cell membrane and organelles. There is cell shrinkage and chromatin condensation, membrane blebbing, nuclear collapse continue and continued membrane blebbing. These are the apoptotic body formations. Apoptotic bodies, they have their own cell membrane and the condensed organelles. Then lysis of apoptotic bodies by macrophages. The mechanism of apoptosis. apoptosis results from the activation of caspases. These caspases they exist as inactive proenzymes that must undergo enzymatic cleavage to become active. So, caspases are involved in apoptosis in the form of proenzymes which are further activated. The process of apoptosis is divided into initiation phase and execution phase. In initiation phase, the caspases become active, and in execution phase, the caspases trigger the degradation of cellular components. After activation, active caspases will trigger degradation of cellular components. There are two distinct pathways involved in caspase activation: intrinsic pathway and extrinsic pathway. The intrinsic pathway, it occurs in mitochondria, and extrinsic pathway involve death receptor domains. So first we are going to study the intrinsic pathway or mitochondrial pathway. It is major mechanism of apoptosis. Mitochondria are remarkable organelles as they contain protein C, cytochrome C, protein cytochrome C that are essential for life. But if released into cytoplasm, it will induce apoptosis. So cytochrome C is an important protein involved in apoptosis. Anti-apoptotic proteins are BCL2, BCLXL, and MCL1. Anti-apoptotic proteins are the proteins which oppose the apoptotic activity or inhibit the apoptosis. They are BCL2, BCLXL, and MCL1. They reside in outer mitochondrial membrane, cytoplasm, and endoplasmic reticulum membrane. They prevent the leakage of cytochrome C. And the pro-apoptotic proteins or factors are BAX and BAK, BAK. So these pro-apoptotic factors, upon activation, they increase the mitochondrial membrane permeability, leading to leakage of cytochrome C and further degradation. BAD, BIM, and BID, these are the sensors. They act as sensors of cellular stress and damage. So in this picture, on the left hand side, you are seeing viable cell, growth factor, and the survival signal. Growth factor, when combined to the receptor, this is the mitochondria and cytochrome C present in it. When growth factor gives surviving signal, there is production of anti-apoptotic proteins like BCL2 or BCLX that will prevent the leakage of cytochrome C. When apoptosis is there is lack of survival signal or irradiation that will cause DNA damage. This will lead to activation of the sensors that will cause antagonism of BCL2. So antagonism of BCL2 is preventing the leakage of cytochrome C. Now, due to antagonism of BCL2, there is activation of BAK channel, which will cause leakage of cytochrome C and activation of the caspases, leading to apoptosis. Now coming to extrinsic pathway. This pathway is initiated by plasma membrane death receptors, member of TNF receptor family. So it is initiated by death receptors which are present on plasma membrane. FAS is a death receptor expressed on many cell types. So the receptor involved in extrinsic pathway is Fas. Fas-L, Fas ligand, is a ligand for Fas and it's expressed on T cells. When Fas ligand binds to Fas, 3 or more molecules of Fas are brought together. When Fas ligand combine to Fas, their cytoplasmic death domain will form Fas-associated death domain. Fas-associated death domain is attached to death receptor, in turn binds an inactive form of caspase 8. The pro-caspase 8 molecule cleaves on another to generate active caspase 8 that will lead to apoptosis. So caspase 8, active caspase 8 is involved in this, leading to apoptosis. This pathway can be initiated by protein flip, which binds to pro-caspase. So when Fas ligand bind to Fas, then Fas-associated death domain is formed, which will activate the pro-caspase 8 to active caspase, leading to apoptosis by execution of caspases. So differences between necrosis and apoptosis. Necrosis is irreversible cell injury due to denaturation of proteins and enzymatic digestion of cells, while apoptosis is a programmed cell death. Causes are hypoxia, toxins, etc. Causes of apoptosis are physiologic or pathologic. Morphology. Necrosis is always accompanied with inflammatory reactions. There is no inflammatory reaction in apoptosis. Necrosis involves many adjacent cells, while in apoptosis, single or few cells are involved. In necrosis, the cell is swollen initially, leading to membrane disruption, and there is damaged organelles. While in apoptosis, there is cell shrinkage, cytoplasmic blebs on membrane, and apoptotic bodies are formed. In necrosis, nucleus shows pycnosis, karyorrhexis, and karyolysis, while there is chromatin condensation in case of apoptosis. Cytoplasmic vacuolations and moth eaten. Cytoplasm become vacuolated and it shows moth eaten. While in apoptosis, dense eosinophilic cytoplasm is there. DNA electrophoresis show a smearing effect in necrosis, while in apoptosis, DNA laddering is seen. Thank you.